Welcome everyone to InBio's latest webinar on cleaning and consumer product testing capabilities, in which we'll be discussing how we can help these companies validate their allergen removal or denaturing claims on their products. I'm Beatty Sturgill, Senior Business Development Manager at InBio. I'm with the lab located in Charlottesville, Virginia, and joining me today will be my colleague, Dr. Maria Oliver, Principal Scientist with InBio, and she is located at our lab in Cardiff, Wales. I'd like to start just with a brief summary of the custom solutions that we're able to provide cleaning and consumer product companies. It's important to start that InBio has significant scientific expertise that we've built on over the past 25 years, primarily in allergen detection and allergen research. Using that expertise, we're able to provide custom solutions for cleaning and consumer product companies, specifically in validating allergen removal claims on their products. This adds a significant value to these products, not only from a commercialization perspective, but in the ability to be able to market these claims. And we're able to help validate these allergen removal claims by using our leading allergen detection technology, which is Maria. Maria is our multiplexing assay in which we are able to detect and quantify the major allergens of clinical relevance from numerous sample types. And we can detect multiple allergens from a single sample. And then finally, we take a stage gate approach when we come up with projects with clients, because we want to be sure we design these projects on a custom basis with our clients so that we can help meet their product goals in validating those allergen removal claims. So that's just a little bit of the background of the solutions we're able to provide. And today, Maria and I will be providing some more in-depth information on this. Maria will start us out with a background on allergens, including common allergens found in the indoor environment, control and removal efforts to date, as well as which allergens you can choose from. I'll then provide some background on our in-solution and surface testing in which we can validate those removal claims. Maria will then take back over to discuss our environmental testing suite, or ETS. This is an advanced facility that is temperature and HEPA controlled that allows an ideal environment for testing allergen products as well as intervention procedures. She'll then provide some simulated use experiments, and then we'll have a quick summary and get into discussion in which we're happy to answer any questions that you have. If you have any questions that come up during the webinar, feel free to click on the Q&A button on the Zoom toolbar, post your questions. You can post them anonymously, and we'll get through as many questions or comments as we can at the end of the webinar. But with that, I'll turn it over to Maria.
Okay, thanks, Beatty. So it's estimated that the average person in the Western world these days spends up to 90% of their time indoors, and the indoor environment can actually be a major source of allergen exposure. So some of the common sources of these allergens might include dust mites, where the dust mite feces actually contain the majority of the dust mite allergens. Allergens can come from pets, such as cats and dogs, although allergens can also come from smaller pets like rabbits and guinea pigs. Then there's allergens from pests, such as rodents and cockroaches. We also experience allergens from pollens from grasses, trees, and weeds. And then there's also allergens that are found in molds and fungi, such as Alternaria and Aspergillus. And exposure to these allergens for asthmatic and allergic individuals can result in many different symptoms: coughing, wheezing, allergic rhinitis, as well as exacerbation of allergen-induced eczema. Okay, next slide, please. So for these asthmatic and allergic individuals, it's been shown that allergen avoidance procedures in their homes can significantly improve the health for these individuals. And this allergen avoidance can come in different forms. It can come in the form of physical barriers, such as anti-allergy pillowcases and mattress encasings, or it can come by the actual physical removal of these allergens from hard surfaces, from soft furnishing surfaces, or from, in fact, the air, with the use of air purifiers or cleaning products such as wipes. But it's important that robust strategies and experimental design is used to provide information on the efficacy of these products. And this is to ensure that they are actually helping allergic individuals in the way that they claim to. So over the next few slides, I'm going to explain here how at InBio we can provide the necessary expertise to do just that. Okay, next slide, please. So firstly, at our disposal, we have two different sources of allergens that we can use for product testing. So InBio manufactures over 100 different purified allergens. These are highly purified. They're single allergens with known concentration and have been highly characterized. So their protein identity and amino acid composition has been verified by mass spectrometry and amino acid analysis, and their immune reactivity has been validated by IgE and IgE antibody binding. Generally speaking, these purified allergens are mostly used for molecular diagnostic platforms. They're also used for in vitro studies and structural analysis, although they can be used for the early kind of benchtop testing for cleaning products. Next slide. But our second option is to use our in-house allergen spiked stock dust. This is a dust that is prepared from normal household dust, but it is spiked with sources of allergens found in their natural form, such as pollen grains or spent house dust mite cultures, for example. And for efficacy assessment of cleaning products and appliances to remove allergens from surfaces or remove allergens from the air, we here at InBio mostly try to use simulated use studies. This is basically where the product or the appliance is used in a way to closely mimic exactly how a consumer would be using it. So in these simulated use studies, our preferred method for allergen assessment is to utilize our in-house allergen spiked stock dust. And this is because it most closely resembles how consumers would be exposed to allergens in their own homes, as well as most accurately reflecting how the allergen removal products or appliances are actually likely to work in these indoor environments. Next slide, please. So another advantage of using our allergen spiked stock dust is that it can be fully customized to contain multiple different allergens within the same stock dust. And which allergens to use is actually a really important decision to make. And we here at InBio can help with this decision-making process. We will get into discussions with you about important points to consider, such as which allergens are the most clinically relevant ones, which are the most prevalent ones, as well as being easily measurable, which allergens will be stable in the dust matrix, and also how easily these allergens can be recovered from multiple surfaces, such as glass, plastic, or fabric. And we recommend testing multiple allergens and not just one or two. And this is because allergens don't all behave in the same way. We often get into discussions with people who try to compare allergen removal with microbial removal, but unfortunately, these don't necessarily go hand in hand. So with most bacteria or viruses, they are basically either dead or alive. With allergens, this isn't the case. Allergens are proteins, but they have many different structures. They have many different functions and properties, and also are found within different forms within the dust matrix itself. Okay, next slide, please. Yeah, as I said, not all allergens will respond in the same way to the cleaning products. So I have some examples for you here. Graph 1 shows some results from a cleaning product on 3 different types of allergens. The black bars represent how much allergen is found in the control stock dust, so nothing has been applied here. The light grey bars, however, show how much allergen remains in that stock dust when a cleaning product was applied to it. So, as I said, we have 3 different allergens. With the dust mite allergen DERP1, we can see that there was a huge reduction in the amount of allergen detected when the test product was applied. This is the same for our pollen allergen BETV1. However, if you look at the cat allergen FeLD1, we see that the application of the product has actually very small or little to no effects on the amount of allergen that's detected. So this just kind of emphasizes why we look at multiple different allergens. Furthermore, even within the same kind of type of allergens, we can see differential responses. So in graph 2, we've taken a cleaning product that was designed for the removal of pollen allergens in general. So for the birch tree pollen allergen BETV1, treatment with the product led to 100% reduction in the allergen. We couldn't detect any of it left. For the Timothy grass pollen allergen, FLEPI5, the product still had a really good efficacy with 97% reduction. But interestingly, for the ragweed pollen allergen AMBE1, the product only had a roughly 15 to 20% reduction in allergen detection. So hopefully this can demonstrate to you why the assessment of multiple allergens is important and how discussions with the scientists here at InBio will help design your studies. And with that, I'll hand back to Beatty to give you some examples of how we can work with you to design the most appropriate projects for your solution and surface testing products.
Thanks, Maria. That's a very helpful background there. So yeah, Maria mentioned, I'll dive in a little deeper on the in-solution and surface testing capabilities that we have here at InBio. So keep in mind that these projects are, again, completely customizable, and that this is just a template that we typically follow that I'll provide. But generally, the projects start with a preparation of the stock dust sample, as Maria mentioned. And these dust samples or this stock dust sample is going to contain sufficiently high levels of the subject allergens that you're interested in your study. With these dust samples, we inoculate it with the allergen source material. Again, it could be pollen granules or spent house dust mite culture, etc. We then sieve it down. This is a picture of our sieve on the right to about approximately 74 microns to obtain a fine dust mixture. We then measure the allergen concentration for, again, those major allergens using our Maria technology. So once we have a well-characterized stock dust sample, we can move into our in-solution testing. And this is where we can test for inactivation or denaturing properties of the solution. So we take the stock dust from part 1 and extract it, and we analyze the product to allergen solution ratios against controls, and the controls typically a PBST solution. The ratios we typically use can be a 10 to 90, 50-50, and then 90 to 10 ratio. We then measure the allergen concentration using our Maria our Maria technology, and we report results as a % reduction compared to those controls. We can then move to our surface testing, in which this is testing allergen reduction or denaturing properties in practice. On the right here, you can see that we are testing, we have our stock dust that we have placed on a hard surface. This is just a hard, non-porous surface that we would use. And we're able to test different surface types using this stock dust. That includes a hard, non-porous surface as seen on the right. We can use ceramic tiles, carpets, textiles, or other fabrics. But what's important about this section is that we follow the manufacturer's usage instructions. So this would be, how do you apply the product? Is it a spray? Are you soaking the material? What's the contact time that you're telling clients of how long to use the product? It could be 5 minutes, 10 minutes, an hour, or until it dries. And also, is mechanical intervention required? Is a wiping required, a blotting, or vacuuming, etc.? But it's important that we follow those manufacturer's usage instructions, because this is where we really want to be able to validate those allergen removal claims. Then we measure the allergen concentration again via Maria, and report results as a % reduction compared to untreated controls. So to provide some case studies of where this in-solution or surface testing can be helpful. So we found with clients that it's very helpful in cleaning solution development. And as I mentioned at the beginning, we take a stage gate approach in which we can share results in real time to help clients change their formulation based on in-solution testing results. Maria mentioned that all allergens are not treated the same. We've had clients find that some allergens, they're not getting the reduction that they wanted in the in-solution testing, and that they've modified their formulation until they can find that balance that they're looking for and the removal that they're looking for. We can also help with the cleaning spray applications. We've had clients change their contact time on how long they recommend a client leaves a spray on a surface to help achieve that reduction percentage that they're looking for in allergens in their validating claims. We can also help with cleaning wipe validations. This would be the ability to remove or capture allergens from multiple surface types. We've also helped with vacuum cleaner validation, and primarily this is used to help validate removal or allergen capture from carpets. And then we can also help with barrier or fabric testing. And for this, we used a modified dust trap system in which we are able to aerosolize that stock dust sample. We aerosolize it through the modified dust trap system, capture it on the fabric or the barrier that we're testing, and then we have a downstream filter in which we are able to analyze using Maria to see if any allergen proteins were able to penetrate the fabric or barrier. And with that, I'll turn it back over to Maria to discuss the environmental testing suite.
Great, thank you very much. So yeah, as well as surface testing, we also conduct testing for allergen removal in the air. And given that the majority of indoor allergens are either permanently or at least periodically suspended in the air, we believe that this is actually a really important area of assessment as well. So at InBio in Cardiff, we have a custom-built environmental testing suite. This testing suite is equipped with a lobby, a dry room, and a wet room. Each of the rooms has independent air handling and temperature control. The airflow is HEPA filtered, but importantly, it can be switched off for experiments that require static conditions to be. Next slide, please. So currently, there's no recognized or certified industry standard methods for validating the removal of aeroallergens. And therefore, at InBio, we've developed and implemented protocols in our own ETS, which simulates a real-life scenario as closely as possible, but still allowing for controlled and standardized experiments to be performed. So for our simulated use experiments of aeroallergen removal claims, we tend to use our 28 meters cubed dry room, which was designed to meet the requirements of AHAM standards. These standards specify particular room dimensions, as well as the number of air exchanges per hour that can be performed, as well as some other particulars, such as surface type and fan position. So the picture you can see here is of our 2 InBio scientists in the dry room setting up for a simulated use test. Next slide, please. So yeah, as I mentioned previously, our simulated use aerosolization tests, we've designed them to be highly standardized so that we can get reliable, reproducible, and valid results. So for all our ETS projects, we always run a background run first of all, just to make sure that the room is clear of allergens before the procedures begin. We then aerosolize our allergen spiked stock dust and keep it airborne with a number of fans. And then we perform our control runs. We then perform our test runs, and our test runs are conducted in a very similar way to the control runs, but in the presence of an intervention this time. And this intervention can be, for example, switching on of an air purifier, or it can be that a cleaning product is being sprayed in the room. And as I mentioned previously, this intervention aims to simulate consumer use as closely as possible. Once we've performed all the control runs and the test runs, all our samples are then extracted and analysed for allergen concentration, again using our Maria assay for multiple allergen analysis. And then the allergen levels, just as in the solution and surface testing, they're reported as percentage reduction in the test samples as compared to those measured in the control samples. And next slide, please. So at the end of the testing, our customers are provided with a full in-depth report. If an appliance, for example, such as an air purifier was tested, within that report, customers will be provided with results in a similar format to the one shown here in the first table. And I have an example here of results for our dust mite allergen DERP1. The background was negative. The average amount of DERP1 that we detected in the control runs was approximately 12.5 nanograms per mil. And the average amount of DERP1 in the presence of the air purifier was just under 2 nanograms per mil, giving roughly an 86% reduction in measurable allergen. If a customer comes to us and would like to test a spray product, for example, we also recommend using a second control here. So as well as our dry control, we also recommend running a wet control. This is important, we believe, because it will basically demonstrate that any differences seen with the product are truly due to the product itself and its specific active ingredients, and not just due to the fact that we've introduced a liquid into the room, bringing the amount of dust out of the air and reducing the allergen levels. So for the example shown here, we have got the addition of the wet control. You can see reduces the amount of that DERP1 by roughly 12% in comparison to the dry control. However, when the product is introduced into the room, here under the title test run, we can actually see that the levels of DERP1 are reduced by roughly 67%. If a customer requires it, we can also provide visual representation of the results, along with any kind of statistical analysis that they would like, as demonstrated by the graph on the slide. However, we'd just like to emphasize that for all our simulated use experiments that we do, InBio is only responsible for providing the results from the product or appliance tested. And any customer product, marketing or other claims that the customer would like to make is due to the customer alone. Unfortunately, we can't make these recommendations if something is used for the allergic individual itself. Therefore, I'll now hand back to Beatty to summarize everything we've discussed today.
Great, thanks, Maria. So in summary, at InBio, we're able to test multiple product types on different surfaces, or as Maria just covered, in air samples. This includes cleaning solutions, cleaning wipes, air purifiers, filters, vacuum cleaners, textiles, etc. We also have a diverse panel of inhalable and food allergens that we are able to analyze for using our Maria technology. And these are the major specific allergen proteins commonly found in the environment, again, most of them having clinical relevance. This includes the major allergens from dust mite, cockroach, cat, dog molds, pollens. One thing we haven't discussed a lot is that we do have a Maria for Foods service line in which we can test the major allergens from foods, including peanut, shrimp, milk, cashew, hazelnut, etc. And we cover the major allergens as regulated in the US and in Europe. We also focus as a real world use scenario. Maria mentioned this at the closing of her slides. But again, the main goal is to validate the allergen efficacy claims by following the product usage instructions of the client's products. And then the results are reported as a percent allergen reduction when compared with controls and untreated controls. And again, we take a stage gate approach. This is a very custom project design with clients, which we maintain communications with during the whole time, and we can check in with results. We can make the client can make formulation changes or product usage instruction changes as we proceed based on those results. And with that, Marie and I'd like to open the floor up for discussion. Again, feel free to post any questions using the Q&A button. You can post any questions there, any comments you have there, and we can spend the next bit going through as many as possible. But with that, we'll turn it over to, let's see what questions we have.